Hello, good afternoon, everyone. Thank you for coming. And I want to start the class by taking up any questions you might have from lecture 8. Does anybody have any requests?
Yes, Professor.
Yep, let me know
I
which one.
have a question. For example, Professor, in the question about B, so Professor, you gave us some percentage, so I did not understand about phenotype, genotype. Can you
Do
please explain
sorry,
about
you don't
B?
understand that B with B?
Yeah, B with the normal wings and vestigial wings.
The B's. Okay, I see. Hold on. Let
Busy.
me pull that question out
Thank you.
and see. Okay, so this is example 4.
Yes, Professor.
Yeah, let me just pull that out. You need me to redo the whole question? Is that what you're saying?
Uh,
Or do you have a
I just did not understand the percentage when Professor you gave each of them 50%. How to make sure they are 50%?
how to
How
make
you how you bring the percentage?
Okay, well, let's just go through the whole question then. I mean, like it's hard to just do part of it and not the other. Okay, so normal wings are dominant over vestigial wings. And again, you don't necessarily have to know what these things means. So we can put like big N for normal and small n for vestigial. And then the other one, the other tray we have over here is the deep pollen sac versus shallow sac. So big D is deep, and then small d is shallow. So we have a male bee over here, a male bee with vestigial wings and shallow pollen sac. Right, so the male, vestigial wings, so little n, and shallow pods, shallow pollen sac, which is little d. And then we have the female with homozygous normal wings and heterozygous deep pollen sac. So for the female, homozygous means the same, so big N, and then heterozygous pollen sac. So far, is that okay? All right, so next we're gonna have to make the gametes. Right, so there's only one type of gamete from the male, which is small n and small d. And then for the female, it's either big N, big D, or big N, small d. Again, I'm assuming that you guys have watched the recordings last Wednesday, right? So over here, we can do the Punnett square. Okay. For the male, here we have small n, small d. For the female, we have big N, big D, big N, small d, like that. So when we combine them, there are only two outcomes here, right? Two boxes. So one out of the two boxes is big N, big D, small d. That's 50%, Nika, because it's 1 out of 2, right? We can say 50% here. And that is normal wings and deep sac. And then the other one, it's also 1 out of the 2. That is also 50%, which is also normal wings, but this time shallow sac. Does that answer your question, or you have a follow-up question?
Professor, just because it is two production, for example,
Yeah, there's
beef.
two outcomes. Two outcomes. Yeah, so
Okay, two
one out
outcome
of the two. Mhm.
or each of them 50%.
That's right. Right. So
Okay.
these are the outcomes, right?
Okay, Professor, the gametes always should be four part or three part.
Not necessarily. It depends, right? Like here, I mean, you can write it four times, but it will just be small n, small d, small n, small d four times, right? So if it's the same thing, I don't have to rewrite it. I only rewrite it once, right? I only write it once.
Why the gamete is Excel and sperm is two, but there's four, five, three gametes.
Sorry, one more time, please.
Professor, you have said gamete is the germ cells, but the
Yeah,
germ cells is just egg cells and sperm cells. But
sperm and egg. That's right. Yeah.
this is gametes. Professor, you provide us many part, three part, four part, but it should
Um,
not be just two.
it should not be just two.
Excel and
Oh, I
pheno.
see what you mean. Yeah, yeah, no, 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 no. What this is saying is for the sperm, right? The sperm, there's only one type of sperm here. They all contain small n and small d. Here, like half the eggs are gonna be big N, big D. The other half of the egg is gonna be big N, small d. That's what it means. These are the types of eggs that can be produced. Yeah.
Okay.
Because for the male, it's only small n, right? So all the sperm's gonna have the small n in it. Do you agree?
Okay.
And the same thing for the D, it's only small d, so all the sperm's gonna have the small d in it. So in the end, there's only one type of sperms. And then for the female, there are two types of eggs.
Okay.
In the
Thank
case
you.
where it's like big N, big D, small d, then you would have four types of sperms or four types of eggs, right? Big N, big D, big N, small d, small n, big D, small n, small d. These would be the different types of gametes, different types of sperms that can be produced as different types of eggs, right? Okay. Does anyone else have other questions you want to take up from the
Y-
study guide?
Yeah, Vincent, I have a question. How the genes are recognized that they are gonna be dominant or recessive?
How, okay, so like we would have to do experiments, right? So for example, if you start with pure breed, and you know, you can assume something is pure breed if it's maintained that way for many generations, okay? So let's say there is like a red and you cross it with another red. Okay? Flowers I'm talking about, okay? If you cross red with red and you do it over and over again, right, for like, I don't know, 10, 20 generations, and all you ever get are just red, and you know, it's pretty sure that these are purebred, right? Because you're not getting other colors. Same thing, you can do that with white, and you can breed it for many generations. And if you only get white, then you can also safely say that the white is purebred. So now we have red that you can be certain is pure, and then you have white that's also pure, and then you cross them, and you get 100% red. That's how you know the red is dominant over the white. If the
Oh,
white,
so the knowledge comes from experience, right?
exactly. Yeah, comes from like doing like just by looking at the gene, like the genetic code, that's not gonna tell you whether it's dominant or recessive. Like you have to examine it. You're gonna look at breeding patterns, right? You have to track the trait essentially.
Thank you.
Okay. Yeah. So there is one thing again, watch the video again. I don't want to explain it. Spend too much time explaining it. The concept again. It's already in the recording. That I told you that there is a mouse, and then one with long tail, and then one with short tail, and then one of the mouse is like fluffy or something like that, and then the other is not fluffy. So this is fluffy long tail, and this is smooth short tail. And then assuming they're pure and pure, and then you breed them, and you end up having a smooth mouse with long tail. smooth, long. Based on this result, which one is dominant? Is fluffy dominant or is smooth dominant?
Smooth.
Yeah, smooth is dominant over fluffy. And what about long and short? Which one is dominant?
Long.
Yeah, long is dominant over short, right? So if you start with traits that are pure and you breed them together, whatever shows up in the first generation, that's going to be the dominant trait. That is key information that you should know for when you do the test quiz. Okay, any other questions? What about these ones, like the study guide question? Do you guys need me to take up any of these at all?
Yes, please. Can you take number 4, the rabbits?
Okay, dihypercross this one?
Yes.
All right, let's do it. In rabbits, black fur is dominant over brown fur. Right, so we should use big B for black, small b for brown. Normal length is determined by dominant gene, and short fur, which they call Rex, is by allele. So you can go like, big N for normal length, and then small n for Rex, short. Determine the diagram across between a homozygous black with homozygous normal length fur. So what is homozygous black? Is it big B, big B, big B, small b, small b, small b?
Big B, big B.
Homozygous black. And then it says homozygous normal length. Big N, right?
Oh, okay. I see where I made a mistake. Okay.
Okay. Well, let's finish it anyways, okay, for the benefit of other people. So big brown Rex. Brown is small b, and then Rex is small n. So this is the P generation, parent generation. So if you cross them, the only gametes you can get from this guy is big B, big N. The only gametes you can get here is small b, small n. So you will get 100% big B, small b, big N, small n. Okay, that's the F1 generation. Any questions so far?
Can you do the F2?
Can I do the F2?
Yeah.
I can. Yeah. So now we take two of these bunnies from F1, and then we cross it with big B, small b, big N, small n. Okay. So you gotta recognize this pattern. This pattern is always going to be, if it's heterozygous for both of them, the pattern, because you don't want to be doing the table all the time on the test. It actually takes a lot of time, right? Here, let me show you the table I'm talking about. Give me a second. It's somewhere here. This table. This is the table I'm talking about. Okay, like we don't want to be doing this table all the time on the test. It's very time consuming. Okay, but that's what essentially that's what the kind of information you need whenever I have heterozygous for both of them. But the key thing to remember, it's always 9 to 3 to 3 to 1. That 9 is the dominant and dominant. Okay, so in this case, black and normal length. The 3s are dominant recessive and recessive dominant. So either brown normal or black and Rex. And then this one is recessive recessive. So if you memorize this pattern, the 9331, and you will use this every time it's like heterozygous for both trait, then you don't have to do the chart. You can just go, okay, 9 out of 16 is dominant, which means it is, in this case, black plus normal fur. 3 out of 16 is dominant, so it's black and Rex. You're gonna have another 3 over 16, which is brown and Rex. No, sorry, brown and normal. Because it's recessive and dominant, right? This is dominant, recessive. This is dominant. And then 1 out of 16 is gonna be brown and Rex, which is recessive. Do you understand? Like, it would save you like a lot of time on the quiz if you don't have to write out this table. Does that answer your question?
I'm still a bit confused, but I think I'll get it.
Confused about what the 9331 is?
No, I understand that it's the addition of the ratios, but I'm still kind of, because I haven't really practiced, so I think that's why I'm still having,
Okay.
a bit of problem. Yeah. So I also have a question on the study guide, the number 6.
Study
I do
guide.
not understand the drawing at all.
Number
The
6.
number 6.
Which number 6? Under which section?
The before the monohybrid problems, just before, yes, the number 6. Examine each of the pedigree below.
Oh, like this one? Oh, did you watch the tutorial video when I take it up?
Yeah, I did.
It doesn't make
This one.
sense?
Yeah. I do not know what we should do here.
Sure, no problem. Let's talk about that. Okay, so the question is asking you to examine. Here, I'll copy one over first. It's to look at this and look at this, and then find out whether this is dominant pattern or recessive pattern. Okay, so first of all, this is the first generation, second generation. Square is a male, circle is a female. That means to get together and have kids. Okay, so this couple has 3 kids, okay? How many boys, how many girls?
2 boys, 1 girl.
Exactly, right? So when it's shading, it means the person has the trait, has the condition that we're looking at. Right? So you look at this and then you have to tell whether it's dominant or recessive pattern. We don't know.
So does the dominant here mean the male?
No. Dominant means the trait always shows up, right? For example, if black is dominant over brown, then heterozygous is going to be black, right? That's what it means. Look over here, right? Dominant, recessive. Dominant means it will always show up, even if you only have 1 copy of it. That's why big B is it's black. Big B is also black. Small b is brown. That's what dominant means. Dominance means it will cover up the other trait. The other trait is not going to show up.
Yeah, I get that. But where do we find the allele in these structures? Because the question says
yeah. So we don't know, right? We don't know. So we can just make it up. We'll just say A is dominant.
Oh,
okay? Yeah.
okay.
Sorry.
Okay.
Sometimes it's good you're asking, because sometimes to me, some certain things are like obvious, but then when you tell me like that, it's like, oh, okay, that's where the confusion is. Okay. Then in the future, I'll make sure to mention that. Okay, so small a will say recessive, right? Whatever it is, like, doesn't matter. Like, it could be hair color, it could be like, earlobe, whatever, it doesn't matter. Okay, but here's the thing, right? This person has the condition. Okay, so like, is the condition that we're looking for, is that a recessive condition or is it a dominant condition? That's the question.
Recessive?
Like, I don't know, right? Let's find out, right? Let's say this is a dominant situation. Okay, let's say I'm just going through the thinking process with you, right? Let's assume it's dominant. Okay? Because we're just guessing. Assume it's dominant. So this person has it, means this must be capital A and something, right? I don't know what the other thing is, but it has to have at least 1 capital A. It's either big A or big A, small a? All of you with me? If we assume this is dominant. Okay. Now, if that's the case, then where does the big A came from? Like, did it come from the father or the mother? We don't really know, but it has to be from one of the parents, right? Let's say it came from the father. But that's not possible, because if the big A came from the father would have the same condition, right? The father would have the same trait, but he doesn't. Well, maybe it came from the mother, but that's not possible either, because it came from the mother would also have the trait, but neither of the parents have the trait. How do I know they don't have it? Well, because they're not shaded in, right? So because of that, it cannot possibly be dominant. It must be
So,
recessive.
Vincent, sorry, I am interrupting you. Can't be the mother like a carrier?
But you cannot be a carrier for a dominant trait, right?
Oh,
Dominant means if you have the allele, you're gonna express it, right? There's no covering it up, right?
okay.
Okay, but you bring out a good point. Carrier, so both of them must be big A and small a. Okay, so this doesn't work, right? It has to be recessive. If this is the case, then they pass it on to the daughter, and that tells you the trait is recessive. Okay, recessive.
So that means the two sons, let's assume, are both capital A's.
That's what they are. A something. I don't know what it is, right?
Okay.
According to the Punnett square, there is a 25% chance that they're going to be big A, 50% chance they're going to be big A, small a, right?
Oh, okay.
Yeah.
So,
And
Vincent, sorry, I have a question.
Mhm.
If this is if this was like um, sex link gene, what happens?
Oh, okay. Yeah. So it's very complicated once you consider sex link versus autosomal. So I'm not going to do that to you on the test, on the quiz. Okay. You just have to think that it is autosomal. It's not related to X or Y. And you just have to figure out whether it's dominant or recessive.
Okay.
Okay. Yeah.
Then should we expect something like this in the quiz?
Yeah, but nothing crazy. Okay, it's just the same similar similar difficulty to the one in the study guide. Okay.
Okay.
In other words, look, and this is the key thing that I wrote down in the recording again. Right, you guys can go check it out. If you see this pattern, must be recessive. If the parents don't have it, when I say don't have it, they don't have the trait. Okay, if the parents don't have dimples, but the kids have the dimple, then dimple must be a recessive trait. Okay, if the parents don't exhibit it, but the kids do, okay, then it's gonna be recessive. Okay. Now let's take a look at the other one here.
What is dimple?
Oh, dimple, like when you smile, the little thing.
Oh, okay.
Yeah. So here's another one. Let's try this. Now this one, right? Do we see this pattern? Do we see parents have it, but the kids don't?
Yeah.
Where?
The parents are shaded and the children, 3 of them are not shaded,
Oh,
and only one of them has it.
but shaded means they have it, right?
Yes.
Yes, but this pattern is the parents don't have it, but the kids do, right?
Mm-hmm.
Yeah, so we actually don't see this pattern, right? We do not see that pattern. In this case, it's the other way around. The parents have it, but the kids don't, right? You understand? It's backwards. Let me zoom out a little bit for you. Over here, parents do not have the trait, but the kid does. Over here, the parents have the trait, but the kids do not. Right? So we do not see this pattern. Right? Let's see what happens. Let's just say, for the sake of argument, let's say it is recessive. If it's recessive, then this is little a, little a, little a, little a, right? That's the only way for you to have the trait. But that's again not possible, because if the parents are little a, then all the kids are going to be little a, right? They all should have the trait, but none of them has the trait. They don't have it. So that tells you it cannot be recessive. This must be a dominant situation. And the parents must be heterozygous, dominant. Right? So the kids are going to be little a. That's the only way for you to have kids that don't match the parents' phenotype, right? So if you see something like this, if you see that the parents have it and the kids don't, daughter or son doesn't matter, this pattern, this must be a dominant situation. Okay? Now, let's just try it out. This last one right here. Just look for the pattern I'm telling you. Do you think this is dominant or do you think this is recessive?
Recessive, I think.
Recessive, yeah. So look, parents do not have it, but one of the kids have it. Same thing here. Parents don't have it, the kids have it. Has to be recessive. Okay, recessive. All right. Let that sink in. Think about it again. Just write out the letters on your own, and see for yourself why dominant would not make sense here. Okay. Another question? Any?
So when parents have it and kids also have it, that's dominant, right?
When parents have it and the kids have it,
Yes.
then that's not conclusive. We have to need more information, right? Because we don't know. If you get a situation where both parents have it and the kids have it, let me explain to you. It could be big A for both, and then therefore big A. But hey, it could be small a as well, right? You see, like it's inconclusive. We don't know.
Okay.
These are the only pattern that would help you definitively distinguish one versus the other. Okay. For something like that, you will have to look for elsewhere on the pedigree, right, to see if there are other information available. Okay. Anyone else from the study guide? If you have not watched the lecture recording from last week, you must. Okay. These are not things you can just figure out on your own. You have to learn the lesson. But any questions from any of these that you want to go through from anybody? Okay, we'll leave it at that then. If you change your mind, if you find something else that you want to talk about, you can always email me. All right. We have a very difficult lesson today, which is on biotechnology. Biotechnology is worse than lecture 8. Biotechnology is actually the hardest lesson of the whole course, I would think. I'll try my best, as I always do, to make it as simple for you as possible. Hopefully you have the notes with you. So let's go through the basic stuff first. The basic stuff are okay. They're not like terribly confusing. Once we move deeper into the lecture, you will see it's heavy. All right, so what is biotechnology? Biotechnology is a compound word. There's the bio, which is biology, and then there's technology. So it's technology that's based on biology. And in biotechnology, scientists are always trying to control, to alter and harness cellular biomolecular processes to help create technologies and products that would help improve our lives. So what does that mean? Give me an example of a cellular process that we've learned in the course so far. What is something that's happening in the cell that we've learned? We learn a lot. I'm just trying to, see if you can give me an example of a cellular process that we've learned. Something that happens in the cell. That's what a cellular process is.
Krebs cycle.
Sure, yeah. So by understanding the Krebs cycle, is there a way for us to control it? Maybe if we could suppress the Krebs cycle in a certain way, then we can create like a drug to treat obesity, slow down metabolism, right? Maybe, okay, or increase metabolism. We understand how DNA replicate. Can we control it? Can we use that to copy DNA that we want to copy? Right? So that's what biotechnology is. You have to understand the cellular process first, then you try to control it, you try to change it and use it to our advantage. So there are many topics in biotechnology. The people who teaching the course, myself and two other teachers, we've decided to go through a few examples with you, starting with what's called recombinant DNA. Recombinant DNA. So recombinant DNA, what does that mean? It means fragments of DNA that is made up of sequences from different sources. Right? So you can take, for example, in this case, you can take a gene from jellyfish. That's what this picture is, right? That's a jellyfish. The gene codes for a protein called green fluorescent protein, GFP. Names are important. GFP makes the jellyfish glow, very pretty. And you can put that into the mouse. These are real mice, okay, that can glow in the dark. So the mouse have recombinant DNA. There's mouse DNA, and then there's jellyfish DNA. Okay. GFP, the green fluorescent protein, has many different applications in medical research. We don't have time to go through all of it. I'm sure you can look it up if you're interested. So I'm gonna show you some examples of recombinant DNA, specifically how it's useful in medicine and in agriculture. Okay, starting with agriculture. There's something called golden rice. Has anybody heard of golden rice before? I'm just curious. Anybody golden rice? Does that make any sense to anybody? No? Okay, so here's the story. Now, vitamin A is an important nutrient for us as humans, okay? Because when a person does not receive enough vitamin A, when they have vitamin A deficiency, that could lead to blindness. Okay? It starts off as like nighttime blindness, and then eventually it could lead to, blindness. And this is actually quite common in poorer countries where they have food insecurity and they don't have adequate vitamin A intake. So that could lower immunity, and combined with low vaccination rate in these countries, it could cause endemic as well as health crises for many different types of infectious disease. So in some countries, each year is estimated to kill vitamin A deficiency to kill 670,000 children under the age of 5, causing an additional 500,000 cases of irreversible childhood blindness. So it's a serious situation, right? And here you can see a heat map that shows you where the vitamin A deficiency is most severe. And you can see it's typically in developing countries, right, with poor access to food sources as well as medical care, right? It's not so much of a problem in North America, for example. So what is golden rice? Golden rice is genetically engineered rice that contains high levels of something called beta carotene. Beta carotene gives the rice like an orangey color, and that turns out to be a building block for vitamin A. Okay. Where else have you heard of beta carotene, maybe? What kind of fruit? Not fruit, sorry, vegetable. What kind of yeah, carrot, right? Carrot has a lot of beta carotene, right? You probably heard the old saying, right? Eating carrot, it's good for your eyes, right? That's where the basis of the story is. It contains beta carotene, which allows your body to make vitamin A. So golden rice was created by incorporating two genes that are related to the creation of beta carotene. There's a gene called Psy, PSY, from daffodil. That's the plant right here. You might have seen daffodils, just in parks. And then there is a soil bacterium that has another gene called CRTL. So scientists extracted those genes and put it into the rice, like regular rice plants, and you have a recombinant DNA. Right? In this case, the DNA contains genetic material from 3 different sources: the rice, the daffodil, and the bacteria, and it allows the rice to produce beta carotene. Okay? So you ship these rice off to places that needs them, and they are much better because it's relatively low cost, and you can store them for long periods of time. And rice is a staple food in many of these places where vitamin A deficiency is a problem. Okay, so the theory is by eating these golden rice that has high beta carotene for it, then your body will make enough vitamin A to correct the problem, right? So there's some controversies. People who support the idea of golden rice say it addresses vitamin A deficiency. It will improve public health. Remember, the deficiency results in reduced immunity, and it could spark endemics, right? And enhance food security. People who opposes the idea of golden rice says that there might be unintended consequences. Like you don't know this is like the oldest argument for people who are against GMO. They're saying you don't know what is it going to do to your body, right? Over long terms, like is there going to be any kind of negative effects, right? You might have yes, you might fix the vitamin A deficiency, but there could be some unintended consequences, some unknown side effects that are bad, right? And people say they don't want corporate controls and patents. Obviously, these rights are created by maybe companies that have access to a lot of funds, and they patent the technology, and maybe they could make money off these things. And they're saying when corporations are involved in these kind of manner, they might have influence on the countries that are using the product. There's also socio-economical factors, right? That needs to be taken into account. So although golden rice is it seems to be a good solution, not everybody is on board. So that's an example of recombinant DNA being used in agriculture and also in healthcare, right? Fixing the vitamin A deficiency. Any questions before I move on to another example?
So when do they do this exchange in the seeds?
Oh yeah, okay. So like they would have to do it in the parent plants, okay? And then you can actually use virus, as you will see, to deliver these genes. And then once you modify them, then the offspring that they produce would have to trade, and all the offspring after that point. Yeah. All right. So next we're going to talk about the case of insulin. So for those of you who don't know, insulin is a protein. You make insulin in the body. Does anybody know what organ makes insulin?
Pancreas?
Yep, the pancreas, which is behind your stomach, okay? Stomach is on the left side of the body, and the pancreas is like behind it. So the insulin is needed to help absorb sugar from the blood. And people with type 1 diabetes they cannot make their own insulin. Like type 1 is the one that usually happens early on in life. It's an autoimmune disease where your immune system attack the cells in the pancreas. It's different from type 2 diabetes, which is the ones that people get when they're older, usually later on in life, tied to obesity and things like that. So for type 1 diabetes, they would need to receive insulin shots. So before insulin shots were available, the primary commercial source of insulin were from cows and pigs until they were replaced by genetically engineered bacteria in the late 20th century. So what's the problem with using insulin from cows and pigs for humans? What are some factors that you can think of that tells you this might not be the best idea? It might not be the most suitable. It might not be the most suitable idea. Okay, let me rephrase. What are some disadvantages of using insulin from cows and pigs for humans?
Because of the DNA difference.
Yeah, could be, right? So like the cow version might not be 100% match for the human version, right?
Yeah.
So that would affect its efficacy. It might not be very effective. It will work, but not optimal, right? What's another reason that you guys can think of?
Lifespan? Like
Of the insulin or of the what? Sorry, what's that?
Of the animals.
Oh yeah, okay, right. So like, you actually have to kill the animals. You have to take their pancreas out, and then you have to extract the insulin from it. So it's
So
animal welfare.
harvest of bacteria is very easier, right?
That's
To
right.
produce.
Yes. So mass producing, that's a problem, right? Like think of how many calves and how many cows and pigs you need, right? To extract enough insulin. Right? So that's another problem, right? And contamination. And also pigs, right? Pigs might not, if it's a pig product, then some people might not be able to use it, right? For religious reason. There's so many things to consider, right? Which is why when you use biotechnology and use bacteria to make insulin, it's much more efficient, much more cost effective, and much safer. So there are two Canadian scientists, Frederick and Benting and Charles Best. They were the one who discovered insulin, and you can look them up if you want. So what I'm gonna go through with you is how to make the insulin, right? And along the way, I'm gonna introduce a bunch of techniques and biotechnologies that allows us to basically mass produce insulin using bacteria. And here are the steps. First, we need to locate the gene of interest. You got to find out where is that gene in the genome. We have 46 chromosome. Which chromosome is it on? And when you figure out which chromosome is on, like which section of the chromosome is it on? Once you figure it out, the location of the gene, you have to clone the gene. You have to make many copies of it. Then you're going to put that gene into a vector. A vector is something that would deliver the gene into the target, in this case, the bacteria. And then you just let nature do the rest. Okay, so this is a petri dish with bacteria growing on them. Some are blue and some are white. The blue ones are the ones that you're interested in this particular case. We're going to use a different method to select them, but we're going to get there. But first, let's start with locating the gene of interest. Like, how do we find out where the gene is? Okay, so locating the gene of interest is much easier nowadays because of the Human Genome Project. That's what this is, right? The Human Genome Project, HGP. That was an international collaborative research program with the goal to map out all the ACTGs in all 46 chromosomes in human. It started on October 1st, 1990, and it was completed in April 2003. So we completed for over 2 decades now. I remember when I was in high school taking biology, my presentation topic was on the Human Genome Project. They just started back then, okay? And now, you know, it has been completed. And they basically map out the entire 3 billion DNA letters. Okay, so it gives us the ability to read nature's genetic blueprint for building a human being. That's basically what it is. So it's available online, and I'm going to show you what it looks like. You can go to something called like the Human Genome Browser. It's on the internet. Okay, and you can search up any gene that you like. Okay, like maybe I'll just go back to the home page here. You can search up any gene that you like. Does anybody remember any genes in the whole course? We learned several. Usually it's a quite a disheartening moment when I ask this question because usually nobody can tell me. Do you remember the name of any gene at all that we've learned? Maybe something to do with cancer. Anybody remember a gene?
Oncogene.
Yeah, oncogene is it's a whole bunch of genes though, right? Do you guys remember like a specific one? Maybe that starts with a B or
Tumor suppressor genes.
BRCA gene.
that's right, BRCA, right? Breast cancer, BRCA1, okay, or tumor suppressor. That's an example of tumor suppressor gene. So you can search up BRCA1 and oops, maybe just go BRCA here, search, and it will show you the genes. It should show you. I'm not sure why it is taking so long. Oh, I think my browser just hang. Okay, never mind. Okay, there it is. Okay, BRCA1 gene. Okay, so then we click on it. Before we click on it, let's see. It's on chromosome 17, and it's right here. Okay. The short arm before the centromere is called the P arm. After the centromere is called the Q arm. So it's on the Q arm, and it's 21.31. That's like the locus, right? I told you. So I can click on it, and then there's a bunch of stuff, but I can get the genomic sequence. It is nucleotide 43,044,295 to 43,125,300. If I click on that, and then I am going to say submit, this is the BRCA1 gene. You see? That's the BRCA1 gene. They map it all out. Right? It starts with, guess what? ATG. Right? What's ATG when you do the transcription?
ME.
Well, yeah.
MET.
AUG, right? AUG, and then it's MET, right?
Yeah.
That's a start codon. You see? It starts with the start codon. Isn't that cool? And, and then it's just pages and pages of these ACTGs. Okay, it took so many years for them to figure out all of these. Okay, so there you
So
go.
it means that this gene is not it's not exist for all women. It exists in some women who are like
Oh no,
in
we all have
danger.
it. We all have it. You have it. I have it. All humans have it. Okay, it's a tumor suppressor gene. What happens is if this gene gets mutated, right? If it changes, if these ACTGs change, then the tumor suppressor gene no longer works. Then those women are going to have a higher risk of getting the breast cancer.
Oh, okay.
Okay, now that brings a good question. Look, this is the normal copy. When I say normal copy, it means like the majority of the population has this one, right? So what they can do, and this is what they actually did, okay? What they did is they collect the breast cancer tissue from patients, and then they examine the DNA of the genes from examine the genes, right, from the from DNA of those tumor cells, and then they look at the BRCA1 gene, because they know exactly where it is, right? It is on chromosome 17, and then they could compare the ACTGs with the normal copy here, and then they could find out where the mutations are. They're like, oh, okay, maybe it's a missense mutation that happens, like, I don't know, I'm totally making this up. Like maybe it happens right here. Maybe the AGC becomes ACC, right? And if you see the same mutation in every single cancer sample, then you can say with confidence, this particular mutation is gonna drastically increase the risk of, breast cancer. Do you understand? Right? It's a very laborious process, as you can imagine, right? That's why research takes a very long time. Okay, so
Vincent, if we take a biopsy for like two different person, so which they have, both of them have like breast cancer, the mutation is the same or the mutation can be different?
Yeah, that's a very good question. Usually it's not a single mutation that causes a person to have cancer, right? And that's the question they're trying to answer, right? They're trying to profile these cancer DNA, basically, right? They're trying to build a library to see what mutations are common in. So, like, let's say we have 10,000 patients, right? If 90% of the people have this particular mutation, then that means it's a very significant mutation. Some mutations are very rare, like maybe 2-3% of people have it. Then that might not be something to pursue in terms of like a therapeutic target, right? Okay, so it's, yeah, like there are going to be shared mutations, but sometimes they could be different as well. Okay. All right. Let's get back to what we were talking about. So that's the genome browser. You can search up any gene. If you know the name of the gene, you search it up. That's what researchers do. They find a gene. Now we know the location. And that's the first step, right? Yeah, first step is to locate a gene of interest. So if you want to make insulin, you're going to find out where the insulin gene is, type it into the internet, it will tell you. Then the next step is to clone the DNA. So to clone the DNA, we use a technique called PCR. And you might have heard of PCR from COVID testing, right? Yeah. The ones you do at home, that's an antigen test. The ones that you have to send out to companies to do, I don't think they pay for it anymore. Like I think you have to pay to get results for PCR tests. Actually, I'm not 100% sure. That's a PCR test, which stands for polymerase chain reaction. So this is the technique that allows specific DNA segment to be cloned or to be copied many times in very short period of time. And you can literally have like millions and sometimes billions of copy within an hour. Yeah, very, very powerful technique. So to do PCR, you need to get a PCR machine. These things are quite expensive, in the thousands. And what you need to do is to set up the reaction cycle. And every cycle, it's going to double the genetic material. So let's say you start with 1 copy. After 1 cycle, you're going to have 2 copies. If you do it for another cycle, it's going to be 4 copies, 8 copies, 16 copies, so on and so forth. So if you do like, let's say 20 cycles, right? You can key that into the calculator. 2 to the exponent 20, that is equal to 1048576. Okay, so after 20 cycles, you already have 1 million copies. Okay, and we rarely start with 1 copy. You can start with like 100 copy, then you will add 2 zeros here at the end. Okay, so let's see.
Vincent.
Yeah.
So once we have gotten the gene,
Mhm.
we want to so are we taking the DNA from the gene now, or what?
Yeah.
DNA?
Yeah.
Okay.
So we haven't taken the gene yet. We just know where it is.
Oh,
Right?
okay.
So imagine
okay.
walking into the library, you gotta find the right book, and then you gotta find the right page. Once you found the right page, now you photocopy that page many times,
Okay.
so you can distribute it to your classmate or something. Okay, so the photocopying technique is called the PCR. Okay, So how does it work? Well, the method is very similar to DNA replication. Okay. So in DNA replication, I hope you remember. I hope you remember something, some of the things I taught you. We use gyrase and helicase, right, to pull apart the DNA strand and to relieve the tension, right? In PCR, instead of using gyrase, we will be using heat to separate the DNA strand. Don't forget the two sides of the DNA are held together by hydrogen bonds. So when you heat it up enough, then the heat will cause the two strands to separate. The hydrogen bond will separate. Okay, and that is around 95 degrees. I mean, it depends. Like if you actually do this in real life, you have to calculate the optimal degree. Because depending on the strand, if there are a lot of A's and T's, then you don't need to heat it up as high. But if there's a lot of C's and G's, then you need to heat it up higher, right? So the heat will separate the two strands, and then the strands will be used as a template to be complementary strand. So in PCR, DNA primers are used to mark the regions we want to copy. Okay, so we're not going to do the RNA primer. We customize the DNA primer. Okay, so in other words, you know, back to our textbook analogy. Let's say we have a textbook. Okay, you figure out the pages. Okay, and then we put these little tags on it. Okay, you put the tag here, and then you put a tag here. Okay, and then you give it to the person who photocopies for you. Then they understand. Okay, I only copy everything between the tag to the tag. Do you understand? Like I put two tags here, right? That tells the person you start this page and then you photocopy all the way until the other tag. That's basically what the DNA primers are. Okay, you don't want to copy the whole chromosome, right? It's like, you know, you saw a million of ACTGs. You only want to copy the section, right? And you know, this thing here, it tells you where to begin, right? This is again from the 43 million to this. So you find out which section you want to clone, beginning and end. Okay, you build a primer to flank it. Okay, let me just get that out of the way. So next we have to have polymerase, but we cannot use regular DNA polymerase 3 because we have to heat the DNA to separate it. We heat up to 95 degrees. If we do that, regular DNA polymerase, they're going to denature. The heat's going to break down the protein and it will not work anymore. So instead, we use something called tag polymerase. Anybody have seen pictures like these before? Do you know what these are? Anyone at all? It's very pretty. Okay, they are natural hot springs, right? They're really hot, hundreds of degrees sometimes actually. And there are actually bacteria that lives in these hot springs. Okay, so scientists have cultured those bacteria and they take the tag polymerase from them. Okay, so those polymerase from these hot spring bacteria can withstand the high heat and not get damaged. All right, so the heat separates it. We have the primers to tell you what section to copy. And then we have the tag polymerase to actually do the copy. So here's the template DNA. We heat it up to separate it. We got the primers. Then we're going to copy everything in between. Just using basic base pairing rule, right? Of course, you have to supply the nucleotides. So let's take a closer look again. Okay. First step is denaturation. Denaturation. So use heat to separate DNA. Once you heat it up to separate the DNA, you need to cool it down a little bit. Okay, we call this annealing. You cool it down to about 68 degrees, 70 degrees, depending, again, like these are very vague number, okay? It depends on the sequence you're trying to clone. And by cooling it, like cooling allows primers to attach. and then you heat it up a little bit again, couple of degrees higher, then this activates tag polymerase. Okay. So you put all of these in a tiny test tube. It's not big at all. It literally is this size. Okay, you put everything, all the ingredients, including the DNA that you want to clone in here, and then you're gonna heat it up to separate it, cool it off to allow the primers to bind, and then heat it a little bit to allow the copy. Then now you have 2 copies, and then you repeat the process. Now you have 4 copies, and so on and so forth. Okay.
Vincent, so when we do this in the test tubes, how do we see when these things are happening? When these, like stages are happening?
How do you know these things are happening?
Yeah.
That's a good question. You don't. Okay.
Oh.
The solution looks exactly the same before and after. Okay. I mean,
Okay.
there are ways we can verify it. And I'm gonna show you some of the techniques later on. But in terms of visually, it looks exactly the same before and after. Okay. Now, listen.
So sorry, Vincent, I have a question. So in reality, when in the cell this replication happens, it needs ATP, right? Like energy. So here, what can help to make this replication?
Yes, we also need to supply energy as well. Okay, so like these are like the core ingredients. There are other things that needs to be added to it, but we didn't go into all the details. Okay, yeah, that's a great question. Okay. It's much easier nowadays. You can almost buy these. It's kind of like cake mix. Okay, everything is already mixed together. It has all the important ingredients. So you just add these pre-made solutions into it, and then all you have to do is supply the source of a DNA that you're trying to copy, as well as the primers. Everything else are gonna be in the premix, so to speak. Okay. So the source of DNA, what's the source of DNA? It could be anything. It could be, just any human cells. Okay. You can give the entire genome, 46 chromosomes, right? Like the whole textbook, right? The whole textbook. Right? But doesn't mean you're gonna copy the whole textbook because of these taggy things, right? Okay. Because of the primers. The primers specify precisely which section of DNA to copy. Even if you supply the whole genome, you will only be copying what's between the primers. You will only be copying the pages between these tags. Do you understand? Okay. So you have control of what to copy. Okay, so we've located a gene, we've cloned it. Now we have to insert the DNA into the vector. Before we do that, I want to ask you a question here. You start with 10 copies of a DNA fragment. If you clone that fragment using PCR, how many copies would you have after 4 cycles? Let's take a look. Okay, I only got 4 answers. Maybe some of you need some help. So 4 cycles, right? Each cycle doubles it. So we can go to. So you start with 10 copies. So 1 cycle, 2 cycle, 3 cycles, 4 cycle. So that's 10 times 2 to the 4. which is 160. Okay. Every cycle doubles all the DNA. Okay. Let's talk about the PCR for COVID tests. The kind of PCR that you do in COVID tests is a special kind of PCR. It's called RT-qPCR. RT stands for reverse transcription, or reverse transcriptase, the enzyme. And Q stands for quantitative PCR. So let's refresh our memory. We start with DNA, and then it becomes RNA, and then it becomes protein, right? From DNA to RNA, I hope you remember, it's called transcription. So it turns out a lot of the viruses out there, they are RNA viruses. Okay? They don't have DNA, they have RNA only. And so when the virus infects the host, they actually sometimes have to convert the RNA back into DNA. So the process is called reverse transcription. Reverse transcription. The enzyme that does that is called reverse transcriptase. Okay? So when you do a COVID swab, the one that you do at home, you swab it, and then you put it into the solution, and then you drop it onto that plastic tray, right? And you wait for the red line to show up. That is an antigen test. That tests for the presence of viral proteins. The PCR test is much more sensitive, and it tests for the presence of viral genetic material. So you swab the nose, okay? You dissolve in a solution, and what happens is you do the reverse transcription. By supplying the reverse transcriptase, you convert the viral RNA, if there are any. If you're not infected, you won't have any, right? So the reverse transcription will convert the viral RNA into the viral DNA. Once you have the DNA, then you do PCR. Okay. So because you cannot detect it, right, on very low level, that's why you need to do PCR to amplify it, to make many, many copies of it. And this kind of PCR is called quantitative PCR. Right? Earlier, you asked me, how do you know you're actually doing the copy? Well, with the naked eye, you cannot see, but there are ways we can detect it. Okay, we can use molecules that are called fluorochromes, and basically they give off light when there are DNA. The more DNA, the brighter it's going to be. So the machine can keep track of how bright the solution is as the copying is happening. Okay, and then you get a graph like this. Okay? This is the color intensity from the fluorochromes, and this is a log scale. It doesn't really matter. Like if you don't understand the logarithm and stuff like that, don't worry about it. Basically, this is the initial copies in the sample, and we're trying to figure out how long is it going to take to cross the threshold. Okay, so like this is like, maybe how long is it going to take to have 100 copies, maybe 100 copies. So we have different samples here. Each of these lines represent a different sample. So let's say, do you think this person is more infectious or this person is more infectious? Is it person A or person B? Right, you get their nasal swab, you do the PCR, and you try to figure out how long it takes to get to 100 copies. Person A takes about 12 cycles to get to 100 copies. Person B takes about 25 cycles to get to 100 copies. So which person is more infectious? What do you think?
A.
Why is that?
Because it takes less time to get more infected.
To get more copies, right? You mean?
Copies, yes.
Yeah. So that means this person already has a lot of viral DNA to begin with. That's why it didn't take too long to make 100 copies, right? Let's say, for the sake of argument, if the person has 50 copies to begin with, it only takes one round for you to get to 100, right? But if someone has, 2 copies to start with, it will take a lot longer for you to get to 100 copies, right? So this person, because the sample is able to reach 100 copy earlier on, this is someone who is very infectious, okay? So this is the person who probably just recently got infected. We got to track the person down and then find the contact tracing, right? I don't think they do that anymore, but this was something that's very important early on in the pandemic, right? So that's RT-qPCR.
Yes, so
Yeah.
is that how they know someone is positive? How it can, how fast it can make the viral DNA copies?
Well, okay. So first of all these people are infected. Okay. If a person is not infected, doesn't matter how many cycles you run, you will never get the DNA, right?
Oh, okay.
Yeah, because the primer would not know where to bind to because the DNA isn't there. Right? But
so the primer is like
DNA.
COVID-19 DNA on its own.
Their DNA, that's right. But you have to design the primer, right? So in this case, the people have to design the primer that would recognize something that's in the virus, but not in the humans.
Okay.
Because when you swap the nose, you might have human cells in there as well, right? So you need to make sure the primer only recognizes viral genetic material. So if you don't have the virus, the primer have nothing to bind to, then there's nothing to clone. There's nothing to copy, right?
Okay, thank you.
Yeah, so the fact that you see a line here means the person is infected. But this also gives you important information in terms of, how infected is the person. Okay, but at the beginning, people were overwhelmed. They didn't have enough testing. So you won't get your results until like 2 or 3 days later. By then, you might have be out and about and, infected a bunch of other people, right? Okay. Just to recap, we located the gene using the online genome browser. We cloned the gene using the PCR. Now we have literally billions of copies of this insulin gene. We gotta feed it to the bacteria. So that's what I'm gonna teach you next. I'm sorry, I'm gonna take away your break again, but I promise I'll finish around 2. Okay. So to understand this next part, we have to know what a plasmid is. A plasmid is a circular piece of DNA that is found in bacteria, in addition to its regular DNA. So these are like the core DNA of the bacteria, and it's not uncommon for bacteria to have extra genetic material in the form of a plasmid. Okay, so these plasmids will give the bacteria special properties. It might allow them to stick better to each other. It might give them antibiotic resistance. And the scary thing about these plasmids is that they could actually be exchanged with other bacteria. Okay, so bacteria A, let's say, is resistant to antibiotic 1. Bacteria B might be resistant to antibiotic 2. But if they come together and exchange the plasmid, now both of them will be resistant to both antibiotic 1 and 2. If this happens multiple times, that's how you get multi-drug resistance bacteria. Superbug, you might have heard of them on the news before. Okay, so plasmids give them special bacteria property. And bacteria, they actually can take up plasmid from the outside. Okay, so we are gonna basically take advantage of these properties of the bacteria. Okay, we're gonna feed the plasmid to the bacteria, but this plasmid is going to contain the gene that we want the bacteria to make. Okay, so in our example, we're gonna have a plasmid with the insulin gene in it, the ones that we copy many, many times. We're gonna put the insulin into the plasmid. We're gonna feed the plasmid to the bacteria, and then the bacteria, not knowing any better, it's gonna start pumping out lots and lots of insulin. Okay, it's gonna do transcription, translation, makes lots of insulin. It's all over the solution. Then we can extract the insulin, right? So that's what we're gonna do. But how do we do that? How do we feed the plasmid to the bacteria? How do we even put the gene into the plasmid? Well, to understand that, we have to learn something called restriction enzymes. Okay, restriction enzymes. Restriction enzymes, they are enzymes that bacteria make. And their job is to cut DNA sequences. We know virus can infect bacteria, right? The bacteriophage that we talk about. So bacteria, they don't like that. So anytime there's foreign DNA coming in, they have these enzymes called the restriction enzymes that will basically cut them up. Right? So they chop up any foreign genetic material. Plasmid is okay because they have plasmid, right? They're not going to chop up plasmid. But if there's any foreign strand of DNA coming in, they're going to cut them up. But they don't cut it up randomly. The restriction enzymes cut the DNA by recognizing very specific site called recognition site. Most of these recognition sites are 4 to 8 base pair long and are characterized by complementary palindromic sequence. What does that mean? A palindrome in English is something that spells the same forward and backwards. So like madam, if you spell madam forward and backwards, it's the same. That's a palindrome. Level, wow, kayak. Okay. This is a very long palindrome. Was it a car or a cat I saw? If you read it backwards, it's the same thing. Okay, so that's a palindrome, right? Now in DNA, palindromic sequences can also occur. Look at the orange part here. Read the strand forward, GAATTC. If you read it backward, it's also GAATTC. So these restriction enzymes, they will find the palindromic sequences and they will cut it. Okay? But each restriction enzyme has its own recognition site, and they are all different. For example, there's something called ECOR-R1. That is a restriction enzyme extracted from E. coli. And the recognition site is GAATTC. You read it backwards, it's also GAATTC, right? Something called small one here, and it recognizes GGGCCC. Okay, and so on and so forth. So each of them has different recognition site. You don't have to memorize them. If a question requires you to know the recognition site, it will be provided in the question. Okay, you don't even have to print this out. So sometimes they will cut zigzag like this. Sometimes it will cut straight down like that. Okay. So the zigzag cutting creates what we call sticky ends. That's actually what they are called. Okay, so that one, they have sticky ends. They are sticky ends because this strand that's like hanging out is complementary to the other side that's hanging out. So you can basically put them back together and they will stick because of the hydrogen bond, right? These ones, they cut straight down. They have blunt ends. For what we are trying to do, we're trying to insert the insulin gene into the plasmid so we can feed it to the bacteria. For that, we want restriction enzyme that create sticky ends. Okay, so the ones with the sticky ends are the more useful one for our purpose. Any questions before we move on?
How do we pronounce the ones with the is it SMA1 or?
Yeah, SMA1 or small one, some people say it. This is ALU1, SOL1,
Okay.
HIND3, that's how you say it. I know it. Like who comes up with the name, right? It doesn't really matter. Right? As long as you understand that they're restriction enzymes, then it's fine. And then they're taken from, these are bacteria name, right? Okay, bacteria name. Okay, so let's take a look. We have small one here, and the recognition site is GGGCCC. So let's take a look at this sequence. I hope you have printed out. Otherwise, you might have to squint at the screen. And tell me how many times you're gonna cut this DNA fragment. If you have this DNA fragment and then you add some small one to it, how many times will it be cut?
Two.
2 times. Okay, so let's see. We are looking for GGGCCC. Oops. And it's 5 prime to 3 prime. So we're just gonna scan the segment and we look for GGGCCC. Okay, this one is CCCGGG. That's not what we want. We want GGGCCC. So we keep scanning. Okay. Do we have GGGCCC from 5 to 3? Okay, we can go look for the other one backwards. Okay. Wait, did I make a mistake here with the 5 and the 3? I think I might have switched them here, so that it's not quite making sense. Hold on, let me just go back to here. 5 GGGCCC. Okay, I think I switched the 5 and the 3. That's why it's not making sense here. Give me a second. Let me just fix this up real quick for you. You know what? I just have to crop this or not. Supposed to be this way. Okay, sorry about that. You just have to change the letters on your sheet, okay? So again, we're looking for 5 to 3 GGGCCC. So right here, GGGCCC. That's a palindromic sequence, and it's gonna cut right there. And if we keep on going, then you're gonna cut it one more time. So essentially, you cut it twice, and then you're gonna have 3 fragments, right? 2 cuts. 2 cuts, 3 fragments. Okay. Sorry about the confusion there. Let's do another one here. Identify the palindromic sequence in the following DNA fragments. And so this one is a little bit harder to do, but I'll try to show it for you. This is GCGC. So if you write the complementary strand, it would be the same backwards, right? You see? That's a palindromic sequence. Okay. Does anybody have another one?
Vincent, so if we want to do this kind of question, so does that mean we have to write the complementary part first before we can solve it?
You can, but you don't, not necessarily, right? So for example, I'm looking for letters that are kind of, you know, repeating themselves. So like over here, you see AATT? What's the complementary strand for AATT?
Oh yeah, okay, it's A8, yeah, TTAA.
Right. And then, you know, I'm gonna check an extra letter just to be safe. So C is gonna pair with G, and G is gonna pair with C, right? So there is the palindrome. Okay. Maybe one more? Anyone at all?
Over here we have TTTAAA.
Yeah, exactly, right. And then you can include a C as well. Okay. So that's all. And there's another one here. Actually, no, just the TATA here. All right. This is just for practice. Like, I'm not going to ask you to identify the palindromic sequence on the test. Like, it's like you can't really circle them, right? So that's that. Okay, last one here. We have the equal R1 and we have the hin D3. So we have the sequence here. This is the whole DNA strand, okay? Like one strand. And we're gonna treat it with the equal R1 and the hin D3. How many fragments will be produced? I'm gonna give you a moment to figure that out. Anyone? All right, let's do it together maybe. For the equal R1, it's GAATTC, starting with the 5. So we're going to find GAATTC. There it is. So that's going to be a cut. It's going to cut zigzag to create a sticky end. There is another GAATTC here. So that's going to be another cut. Another cut right here. And that's it. That's the cut from equal R1. Let's see about Hindi 3. It's AAGCTT. So AAG, just gonna scan for the palindrome. AAGCCT is right here. Okay, and then we're gonna have another cut. So total of 3 cuts. 3 cuts is 4 fragments.
It means like they cannot do overlap. I mean,
they are not the same though, right? So like there wouldn't be any overlap. Okay, even though this looks like it's AAG, right? But then it's not followed by CCT. So it's not gonna, is that what you're asking, or?
yes, thank you.
Okay, so we will finish the note next time. This is gonna take like a good 20-25 minutes. We don't have time for that today. No quiz this week, but you will have one next week, which is, let me just double check when is it, so I don't give you the wrong information. The quiz is gonna be next Monday. Yeah, next Monday. Okay. I'll see. Okay. I'll have to finish the biotechnology on Wednesday. Maybe I'll move it to the Wednesday to give you guys an extra 2 days to prepare. And then just 2 more weeks and we're done. Okay. That's all. Any other questions?